Hello, welcome to Module 5, Chapter 9. This video is going to be on the resting membrane potential. It's a third in, I think I have 7 short videos for Chapter 9. And this is the third time that I have recorded this because for some reason the PowerPoint file keeps getting corrupted. So let's try it. Third time's a charm. Okay, so get my pen. This is a cross section of the spinal cord, and coming out of the spinal cord, this is the ventral region of the spinal cord, and then this is the dorsal region of the spinal cord. And coming out of the ventral region of the spinal cord are these two neurons. One is a red neuron and one is a purple neuron. And they come out of the ventral region of the spinal cord, and then they travel to the muscle cells. And they travel in a nerve. So this yellow thing is cut here. It's a nerve. The neurons, which are nerve cells, travel in the nerve, and they go out and they innervate these skeletal muscle cells. So each one of these, this is a skeletal muscle cell that's cut. So there's, let's see, 1, 2, 3, 4, 5 skeletal muscle cells in this cartoon. And the neurons, the red and purple neurons, they start here in the spinal cord. They have a cell body, and then they have this purple and red thing called an axon. And the axon is just an extension of the neuron cell body. The axon goes out and makes contact with a muscle cell. So these muscle cells do this setup. They possess different characteristics. The first one is irritability. That is the ability of a muscle cell to receive and respond to a stimulus. So the stimulus being the electrical signal and neurotransmitter that comes from this motor neuron. Muscle cells also possess the characteristic of conductivity, so that's the ability of a muscle cell to transmit electrical impulses, aka action potentials. Muscle cells also can contract, so they can get short and get shorter as a function of the sarcomeres getting shorter. Muscle cells also possess the characteristic of extensibility, so they can be stretched, and then they can also recoil, so muscle cells are elastic. So this is our first foray into electrophysiology and the resting membrane potential. So here's our neuron. This is the neuron cell body, so you'd find this, if it's a motor neuron, you'd find this cell body in the spinal cord. And then here's the axon that would go out, and this axon is branching, and then these terminals would innervate a skeletal muscle cell. And then you take a piece out of the axon, and you can see that this part, this yellow part, that's the plasma membrane of the axon. Here's the other part of the plasma membrane of the axon. And then here's inside the cell. This yellow stuff is inside the cell, and the blue stuff is outside the cell. So outside the cell is the ECF, or extracellular fluid. Inside the cell is the ICF, or intracellular fluid. And then you can see you have concentrations of these ions. So we'll look at potassium first. So the concentration of potassium ion is higher inside the cell than it is outside the cell. The concentration of the sodium ion is higher outside of the cell than it is on the inside of the cell. And the concentration of the chloride ion is higher outside of the cell than it is inside of the cell. And then also inside the cell you have concentration of these large anions, so big molecules with negative charges. And these guys actually can't leave, the A-, the anions, can't leave the cell. So with this setup and all these concentrations, you find the inside of the cell is negative relative to the outside of the cell. So you see all these little negative signs on the inside of the cell, and then you see all the positive signs on the outside of the cell. That shows you that the relative concentration of negative ions is larger inside than it is outside the cell. And then, so you take this little piece here of the plasma membrane of the axon and you enlarge it, and you see these arrows. So it shows, this arrow says potassium diffusion, this arrow says sodium diffusion, and this purple thing is the sodium-potassium pump. So if there is a potassium channel open, potassium will diffuse down its concentration gradient because it's higher inside the cell than it is outside the cell. If there is a sodium channel with the sodium and the gate in the sodium channel is open, sodium will move from the outside of the cell to the inside of the cell down its concentration gradient. And then the sodium-potassium ATPase is in every cell of your body, and it's always working using ATP. It pumps 3 sodiums from the inside of the cell to the outside of the cell, and it pumps 2 potassiums from the outside of the cell to the inside of the cell. So the resting membrane potential then is an electrical gradient. So gradient, you have positives on the outside, negatives on the inside, between the ECF and the ICF. And the plasma membrane, this yellow thing, acts as an electrical insulator. So it's the resting membrane potential, it's called the potential because if you remember, potential energy is stored energy, and because you have charges on either side of this plasma membrane that's acting as an electrical insulator, you're storing charge. So in the resting membrane potential means that the electrical gradient isn't changing much. So you're mostly staying negative on the inside and positive on the outside of the cell. Potential, from potential energy, means that that plasma membrane stores potential energy in the form of an electrical gradient between the ECF and the ICF. So if you open a channel and positive charges go in or negative charges go out or vice versa, then those ions, those charges are moving, and you go from being potential energy to kinetic energy. The electrical gradient at the plasma membrane is mostly determined by the potassium gradients because there's a lot of different potassium channels in a plasma membrane. So for example, there's potassium leak channels where potassium is slowly leaking out of the cell. But then the sodium-potassium ATPase will pump potassium back in the cell. There's voltage-gated potassium channels. So we'll talk a lot about channel physiology, the different types of sodium and potassium channels. So this is just another cartoon of sodium-potassium ATPase, this big purple thing. For every ATP that connects with the sodium-potassium ATPase, you pump 3 sodium out against its concentration gradient, and you pump 2 potassium in against its concentration gradient. And what I mean by that is because sodium has a large concentration outside of the cell, and sodium has a small concentration inside the cell. Just do that by making big and small sodium symbols. If you want to get sodium to go from a low concentration to a high concentration, that's moving against its gradient. So you have to do that with a pump, and you have to spend energy to do that. So that's what you do with ATP. And it's the same for potassium. If you want to pump potassium in the cell, because remember, potassium is really large concentration in the cell, and potassium is a small concentration outside the cell. If you want to pump against its gradient, you have to use sodium-potassium ATPase. So the pump allows the ECF to build up more positive charges and the ICF to have more negative charges. And this just tells you this fourth square says that sodium has a gradient. It wants to move down its concentration gradient and its electrical gradient inside to go inside the cell. So up here in the right-hand corner, this is just a cutout of the plasma membranes, the phospholipid bilayer, and that's what these two lines are going to represent, the phospholipid bilayer. And in the muscle physiology world, they call the phospholipid bilayer, aka the plasma membrane, they call it the sarcolemma. And it just shows that positively charged cations are in the ECF and a high concentration of sodium, and in the ICF, inside the cell, negatively charged proteins and a high concentration of potassium. So I told you those large anions, negatively charged molecules, don't leave the cell. They're too big. You have the sodium-potassium ATPase in the plasma membrane that pumps sodium out and potassium in. So the sodium-potassium pump and the potassium leak channels create an electrical and a chemical concentration gradient. So it's a concentration gradient. We just talked about that because if sodium is high on the outside and low on the inside, if there's a sodium channel open, sodium will go from the outside to the inside down its concentration gradient. But remember, the inside of the cell is also negative with respect to the outside of the cell. So if you are a positively charged ion like sodium, then, and there's a channel open with the gates open, let me make the gates open there. Okay, there's the gate. If you are a positively charged ion like sodium is, it's going to be attracted to that negative interior of the cell. So sodium is going to move down its concentration gradient and its electrical gradient if sodium is permeable to the cell. So that permeability means there's an open sodium channel. Okay, so we've set this up. Now, if we want to make a graph of what it looks like to have the resting membrane potential and when sodium and potassium ions move in and out of the cell, we want to look at a graph of that. We take this thing here, this is a probe, and so the probe would be connected to a voltmeter, and the voltmeter would read the voltage inside the cell in millivolts, and usually in a neuron, motor neuron, the negative inside of the cell measures about minus 90 millivolts. Okay, so here's a question that you guys can try to do this. Get a piece of paper out. If the sodium channels in the plasma membrane open, so let's say some sodium channels opened, the channel was open, the gate, the sodium gate was also open, and you have a large concentration of sodium on the outside, and you have a small concentration of sodium on the inside. What I want you to do is sketch and explain what would happen with sodium. So the channel's open, the gate's open, what's going to happen with sodium, and if you were to draw a graph with millivolts on the y-axis and time on the x-axis, and let's say you start your resting membrane potential at minus 90 millivolts. So this is your resting membrane potential. And now all of a sudden, right here, you open up sodium channels and open up the gates as well. What's going to happen? And let's say you're more positive up here. What will this graph look like? So turn this off and draw it, and then we'll see what it looks like. Did you draw it, or did you just go to the next slide? Well, here's our graph of the membrane potential. So at A, remember that's the resting membrane potential. And in this cartoon it says minus 70, but it could be minus 92. It just depends on the cartoon you get. Membrane potential in millivolts is on the y-axis. Time in milliseconds is on the x-axis. If you open the sodium channels, tons of positive sodium ions are going to pour inside the negative interior of the cell, and that negative intracellular fluid is going to become more positive. So you would draw it like this on the graph. So this part of the graph is when sodium is rushing into the cell and making it more positive. And this phase is called the depolarization phase of an action potential. So now, if sodium channels close, so now the sodium channels are closed. So sodium can't get in anymore. But potassium channels open, and their gates are open. Sketch what would happen to sodium. Could it go in anymore? And then what would happen to potassium, and sketch the same kind of graph we just did. Okay, so if sodium channels closed, it would be here. So sodium channels closed at the top. That means no more sodium is rushing into the cell. Now, potassium, because it's larger concentration inside a cell, and the potassium channel is open, so the membrane is permeable to potassium. Potassium is going to leave the inside of the cell. Positive charge is leaving the inside of the cell. That means that the millivolts are going to become more negative. And they actually do a little bit of an undershoot, and then they come back up here to resting membrane potential. And this is a graph of an action potential. This is actually what happens all along the plasma membrane of a muscle cell as an action potential travels along a muscle cell. So muscles, we looked at this cartoon before, so we know this is the cross-section of the spinal cord. And we have a purple motor unit and a red motor unit. So a motor unit is just one motor neuron, so the purple motor neuron, one motor neuron, and all the skeletal muscle cells stimulated by that motor neuron. So the motor neuron, the purple motor neuron, it branches here, and it innervates one, and it comes down here, it branches again, innervates 2 and 3 muscle cells. And then the red motor unit, the red motor neuron of the red motor unit comes out, and it branches here, and it innervates 1, 2 muscle cells. So the purple motor unit is larger because its motor neuron, its purple motor neuron, innervates more muscle cells. And a motor unit can have hundreds of muscle cells. It only just shows innervation of a few muscle cells in these cartoons. And then here's all the cartoons is filled in with all the descriptions. And then, so the neuromuscular junction then is the site where the axon terminal, so here, that the purple motor neuron, its axon terminates, it's called the axon terminal, and it's going to meet the sarcolemma, or the plasma membrane of a muscle cell. And then there's a couple things from Marieb, the textbook that we use, and you can read over that, and that's it.